had a very distinguished academic career and was a professor at Southwestern and did a lot of work with immunology and bioinformatics. Had a big focus on influenza, where I think he's been the curator or the maintainer of the influenza immune database. And he came to the J. Craig Ventnor Institute as head of bioinformatics, collaboration with people at the Supercomputer Center here, and has more recently become the director of the whole building. I don't know what that means, but it probably means less free time.
Yeah.
And I really got to meet Richard with regard to the human vaccine program, which is an initiative to really characterize the human immune response to help develop vaccines. And one of the things he'll talk about is a project as part of that. And he also has done some incredible work in other areas, and I thought his brain work is sufficiently interesting that everybody would want to hear about it. So he can tell us his expertise in both wet lab activities and bioinformatics. Richard, thank you.
Well, thanks for the invitation, Doug. Yeah, I'm actually an immunologist and an infectious disease person, but I'm going to tell you about a project that we're focused on looking at human brain. I kind of got dragged into this project because of our expertise in bioinformatics and computational biology, but it ended up being a fascinating project, and hopefully you'll appreciate that. And I will try to touch at the very end on immune responses, but the majority of the talk is going to be characterizing human brain. So we've been very interested in using single-cell genomics, and the main reason that we're interested in that is that, I think it's pretty clear that cells are really a fundamental functional unit of multicellular organisms, and obviously different cells play different roles in the body. We all know that. And really the function, the phenotype of the cells is really dictated by that subset of genes that's expressed in a given cell at a given point in time. And so any kind of abnormality in the expressed gene list will give rise to disease. And we can use those kinds of differences in gene expression as diagnostic tools. Historically, we looked to try to understand the cellular diversity using techniques like transcriptomic analysis, but we were always doing that using bulk samples. And trying to understand the cellular complexity in bulk samples is obviously very challenging because you have a mixture of different cell types. They're present in different levels in the mixture. And so it can be very difficult to look at the rare cell subsets and figure out what they're doing. And so with the advent of single-cell RNA sequencing, this has really allowed us now to quantify cellular phenotypes in a very unbiased fashion, and allows us to look at both known cell types as well as novel cell types in these complex mixtures. And then finally, we have taken advantage of the use of artificial intelligence and machine learning to help us interpret this kind of single-cell RNA-seq data. So at JCVI, we have a Center for Single-Cell Genomics, and we've got a number of different projects that are ongoing there. Most of the projects either involve looking at human brain or looking at the human immune system, either looking at vaccine responses or autoimmune diseases. And we think that single-cell genomics is really a powerful platform to help us analyze these different kinds of systems. So what I'm going to focus on today is these kind of first three topics here. We have just completed an analysis of one region of the human brain, middle temporal gyrus. I'm going to show you the different types of cells that we've discovered in that region. But the focus of my talk is really going to be on some of the computational methods that we use and that we developed to help us interpret this single-cell RNA-seq data and really characterize the cellular complexity of brains. And I think at the end, I have a couple of slides on looking at immune cell subsets, just as another example. But this got started with our involvement in a large international consortium called the Human Cell Atlas Project. This is being led by Aviv Regev at the Broad Institute and Sarah Sanger in the UK. And the idea for this project is to try to come up with a comprehensive collection of all the different types of cells that are present in the human body, in all the tissues in the body. And the initial focus of the Human Cell Atlas is on healthy tissue. And so the idea is that then this would provide kind of a foundation for doing any kind of comparison of different disease states and disease tissues. And there are really two kind of main branches to the project. One is to do kind of single cell transcriptional profiling of isolated individual cells. And then the other is kind of a spatial branch, that's spatial transcriptomics, to understand, you know, the geographic location of different cell types and how they interact with each other in tissue. And this project involves several hundred people in a variety of different institutions around the world. But we've been focused on the human brain. And so the work that I'm going to tell you about today started out with looking at one region of the neocortex, the middle temporal gyrus here, which is involved in some kind of higher order functions like speech recognition and things like that. It's been quite clear that there are different types of neurons in the human brain. And this is kind of a classic diagram that you find in the textbooks, where you can see that neurons were defined based on morphology. This is a pyramidal neuron, a projection neuron. The axons project into other regions of the brain. And it has a certain kind of triangular shape. But then there are other neurons that have different types of shapes. And these inhibitory neurons up here are kind of more horizontally situated versus these pyramidal neurons, which are more kind of vertically situated. So we knew that there were likely to be different types of neurons based on these different morphological characteristics. But what we wanted to do is we wanted to kind of take a broad approach and apply a whole variety of different experimental platforms to neurons in the brain to try to further characterize their phenotypes, using transcriptomic profiling, characterizing their anatomical structures, performing electrophysiological measurements on isolated individual neurons, and also doing kind of a synaptomic analysis, figuring out which neurons are touching which other neurons and how that connectivity works. With the ultimate goal of trying to produce a gold standard taxonomy of neuron types in the neocortex. So to do that, we've been doing single cell transcriptional profiling as kind of our first major high throughput technology. Now, it turns out that when we first tried to do single cell RNA sequencing, we weren't successful, because if you think about how the neurons are kind of interdigitated in the brain, when you try to liberate single cells, they end up getting disrupted. And so you end up with just a bunch of debris. It's very difficult to isolate intact neurons from brain. But it turns out that with just gentle detergent, you can float the nuclei out of the cells, out of the tissue. And the nuclei has the same kind of transcriptional profile as the whole cell, except that we get about 1/5 of the amount of RNA from a nucleus than we would get from the whole cell. And so the work that I'm going to tell you about is actually single nucleus RNA sequencing that we're doing. So this is the middle temporal gyrus in a brain slab. The cortex is divided up into 6 regions that have different characteristics, and you can identify these regions with a Nissl stain. And so what we did in this experiment is we're kind of interested in which cells are located in which regions. So we microdissected out these 6 different cortical layers. And then using Downs homogenization and mild detergent, we can generate a single nucleus suspension. And then there's actually a marker that's expressed on the nuclear membrane of neurons, nu-n, and we can use this to sort the nuclei based on whether they're neuronal or non-neuronal. And so we sort out neuronal and non-neuronal cells into microtiter plates, and then we use a single-cell RNA-seq protocol called SMART-seq to do the amplification and generate the libraries that then go into Illumina sequencing. And so the experiment I'm going to show you, we ended up with about 16,000 single nuclei that we sorted into microtiter plates and processed through this SMART-seq protocol to look at their transcriptional profiles. So this is a schematic of the SMART-seq2 protocol, and really the special sauce in this technique is that it's designed to specifically enrich for full-length transcripts. And the way that it does that is using this so-called template switching oligonucleotide. So what happens here is, here's the oligo-DT primer on the poly A stretch of the messenger RNA. This is extended by reverse transcriptase. But when RT gets to the end of the template, it adds on a few non-templated C residues. And so what we do is we add this second oligo in here that has a poly G tail to it. And that allows, this kind of anneals to that non-templated C stretch. And then it allows us to extend that first strand cDNA a little bit further. And now we can use PCR amplification using the single shared primer on the two ends to amplify this molecule. And because we're kind of requiring it to have this poly-C stretch here, it enriches for full-length transcripts. And so this technique works really well, and it's really one of the workhorses now in the single-cell RNA-seq field. So once we generate the libraries, do the sequencing, we process the primary read-level data, and align it to reference databases, reference genomes, and estimate the amount of transcript per gene. And that ends up giving us a cell by gene matrix. So this is an expression matrix where each column here represents an individual nucleus or an individual cell, and each row is a gene, and this is the relative expression of that gene. And so typically what we do then is we take this expression matrix and we cluster it so that we're basically grouping together cells that have similar expression profiles. And this is kind of what a clustering result might look like. And the hypothesis is that if these cells have a very similar transcriptional profile, they're likely to be cells of the same type. And so these expression clusters we consider to be equivalent to cell type clusters in this kind of analysis. So when we did that with the 16,000 nuclei from middle temporal gyrus, this is what the clustering results look like. So we ended up with 75 distinct transcriptional clusters. This is kind of, they're color-coded based on the cluster identity. And I'll kind of come back to what these different clusters represent. But we had samples from 8 different specimens. So 6 of the specimens were post-mortem frozen brain tissue, and 2 of the specimens were fresh epilepsy biopsies in a part of the brain region that was not affected by the lesion. And what you can see here is we've color-coded the nuclei based on the specimen source, and there's really good intermixing of nuclei from these different specimens in all of the different cell type clusters. So we're not seeing batch effects related to the specimen. It looks like we're getting a good mixture of nuclei from different specimens in each of the clusters. And then this is the same clusters color-coded based on the cortical layer that were micro-dissected. And so you can see in some cases you have clusters like this that are enriched in a particular layer, layers 5 and 6, so deep in the cortex. And then you have other clusters where there's kind of a mixture of nuclei from the different layers. So all of this looks pretty good. So the first thing that we did was we looked at the expression of certain well-characterized marker genes for neurons to see how those marker gene expression patterns map to these clusters. So this is a hierarchical cluster of clusters. So each one of these columns represents one of those clusters that we saw in that PCA plot, Tisney plot. And so here are some well-characterized marker genes that are known to be expressed in different types of neurons. So first of all, here you can see this is expression of GAD1, which is a marker of inhibitory neurons. So you can see that all of these cell type clusters express GAD1. And so that marks these as GABAergic inhibitory neurons. The other main neuron type are excitatory neurons that express, that are glutamatergic, and they express this marker SLC17A7, and that's these guys over here. So these are all the excitatory neurons. And then over here are a set of non-neuronal cell types. So these are the glial cells in the brain, microglia, oligodendrocytes, astrocytes, etc. So within these major classes of inhibitory and excitatory, you see also other markers that are known to tag subsets of neurons like VIP1, somatostatin, this LHX6 gene. And so what we're seeing here is a pretty good recapitulation of known biology in terms of marker genes that have previously been characterized to be expressed in certain types of neurons in the brain. But obviously there's additional complexity in comparison to this kind of prior knowledge of these major cell subsets. So what we wanted to do then is dive into this data further and try to characterize the transcriptomes. And specifically what we wanted to be able to do is see if we could come up with specific markers for each one of these cell types to figure out which genes are differentially expressed in each one of these cell type clusters. And so that's where we decided to take a machine learning approach to help us identify these markers. So this is work that was done by Brian Averman in my group, and we're using a technique called explainable artificial intelligence to do this. So the reason that we want to identify marker genes is that they can be really useful for a lot of our downstream applications, especially the spatial transcriptomics part of the project. We want to be able to use fluorescence in situ hybridization, multiplex FISH, to be able to figure out where are all these different types of cells located and who's located next to whom. And so in order to do that, we want to have very specific markers for each of the cell types and the minimum set of markers, because we're limited by the number of fluorescent reagents that we can use in a multiplex FISH assay. These marker genes also are pretty useful for quantitative PCR targets. In the work that we're doing in immunology, we can use them to identify good flow cytometry markers for cell sorting. We can use them computationally to do deconvolutional analysis on bulk RNAseq. And we can use, and I'll show you how we're also using these things as reference expression matrices for comparative analysis. So, these marker genes are useful for a variety of different purposes. But what we wanted to do is come up with this minimum set. So we decided to use machine learning for that, and there are a variety of different flavors of machine learning algorithms that one could consider using. And this is just a kind of a partial list of different methods that have been developed that fall into this category of machine learning methods. And you can kind of characterize these methods based on two properties. One is the prediction accuracy. So you develop a predictive model using these methods, and then you can test that model on an independent data set and figure out how accurate are your predictions on the new data set. And also on explainability. So not only do you have this model that can do this prediction, but how explainable is this model? how do we know that the model is working? What kind of features are useful in solving the classification problem? So decision trees are relatively simple machine learning models. They are really great with explainability, but they're not very good in terms of prediction accuracy. So this is an example of a decision tree. So here you're basically asking a series of questions, and based on the answers to those questions, you have a result, yes or no. And we know exactly how we arrived at this decision because we keep track of these questions that are being asked. And so this has a high level of explainability. We know how this model works. But if you try to apply this to a new data set, sometimes it's not very good at predicting how a new data set is going to respond. And then in contrast, you have things like these deep learning neural networks that are generated by doing kind of a weighted combination of a variety of different features that are then passed through multiple layers to arrive at a decision result. And the problem is it's impossible to interpret these kinds of very complex neural networks. So they're not very good at all in terms of explainability, but they tend to be quite accurate in terms of their prediction accuracy. So, but it turns out that there's one method that we use, have started to use routinely in our group that seems to give us the best of both worlds. And that method is called random forest. And so random forest retains a fair amount of the explainability of these decision trees, but also gives us good prediction accuracy. So I'll show you kind of how the random forest works using that gene expression matrix that we want to use to pull out these marker genes. So basically, this is, let's say here we have 4 different cell type clusters. What we want to do is identify a gene whose expression allows us to segregate these different clusters of cells. And so, for example, here, if we were to pick the expression of this gene at this level, it's really good at segregating these 2 cell type clusters from these 2. And then we can look at a different gene and partition this set here, and now we get clean separation of both of these clusters. So the expression of these 2 genes would be quite useful in helping us develop a classification model of these different cell type clusters based on the expression of those genes. So the way that random forest works is that we don't just feed the algorithm all of the data, we actually subsample the data. We subsample the cells and the genes. And then we ask the algorithm to, out of this subset of genes and this subset of cells, which are the best genes in partitioning the data to segregate these different classes. And that gives us a decision tree here, and it selects out the genes that are most useful in these branch points in the decision tree. So we do that once, and then we keep that tree, and then we start over again, and we pick out a different subset of genes and a different subset of samples, and we iterate that process over and over again. And that generates a forest of decision trees. So this is where the random forest comes in. Here's the forest, and we're randomly sampling the data. And so typically we'll do this about 10,000 times. So this is now our random forest model. It's this 10,000 decision trees. And what we do is we can take new data, run it through all 10,000 decision trees, and just have it, through a simple voting mechanism, have it tell us what class this unknown sample belongs to based on how it's processed through this forest of decision trees. So that turns out to work pretty well in terms of prediction accuracy. But what's important for our use is that as this algorithm is running, it's keeping track of the features that are most useful as we're building those decision trees. So this is an example of what you get a ranked list of features that are useful through this process. And so here's an example. This was a gene, this is the first gene that was useful in segregating one of the excitatory clusters, E1. And it was available as a feature in 18,089 trees from that random sampling process. And when it was available, it was selected as the first feature, the most useful feature, in 18,086 of those trees. So when this gene is available, it is almost always selected as the best feature for that first decision point, that first split point. So we end up having this really nice ranked list of useful features that also comes out of the random forest method. And this is where the explainability is for those models. So we were interested in coming up with a minimum set of marker genes. And so the way that we do that is we take this expression matrix, we generate the random forest, that gives us a ranked list of useful genes. And then what we did in the first version of this algorithm that we call NS forest, we generated a decision tree with the first gene, then the first 2 genes, then the first 3 genes in the list. And then we basically measure the classification accuracy using just the subset of genes from the ranked list. And we stop selecting new genes when we reach a maximum in terms of classification accuracy. So that worked reasonably well. And this is an example of what one of these decision trees look like. So here you can see we had 299 nuclei in the E1 cluster, and then there were 572 in all of the other clusters, so that's the non-E1 group. And if we use the expression of TESPA1 and we have a threshold of 23 units, and we do that segregation, you can see that now over here in this group, 294 of the E1 nuclei come over here because they express high levels of TESPA1, and only 7 of the non-E1 group go in that direction. And over here, 5 of the E1s are over here, and almost all of the non-E1s are over there. So you can see why this is a good gene for distinguishing between those classes. So we applied this to data just from the layer 1 of the cortex. And this is the set of marker genes that came out of this analysis. So there was something like about 27 marker genes were required to identify the 16 clusters from layer 1. And so here's an example. 3 genes gave us maximum classification accuracy for E1. Here's TESPA1. We got a little bit better if we included this SLC17A7. And then the absence of expression of this other gene was also useful in giving us maximum classification accuracy. So in this case, we get both positive and negative markers. And then also some of the markers were quantitative. So this KIT gene, for example, is expressed at really high levels in this cluster, but it's also expressed in other clusters, but at a lower level. So it turns out that while we were getting good classification accuracy with this, those 2 features were not ideal for our downstream applications. So for example, if you're doing in situ hybridization and you're looking for the absence of fluorescence in regions, that's not very good. That's going to be problematic to try to interpret those results. And also these quantitative markers are also not very good in fluorescence in situ hybridization. So we decided to, this is what we do in our group, is we build new algorithms. So we took that random forest algorithm and now we modified it to try to focus only on positively expressed genes and genes that show a binary expression pattern. And so we take the top ranked genes and we developed a binary score so that it's expressed in the relevant cluster and not expressed at all in the irrelevant clusters. So we basically are re-ranking it, ranking the list based on this binary score. And then instead of going first gene, first 2 genes, first 3 genes, we do all combinations up to 3 genes and then pick the one combination that gives us the highest classification accuracy. So this is the version 2 of the algorithm. Do you want to ask a question?
If you dial down the complexity of the random forest by limiting the number of samples, when do you, because we're concerned about sample number, when do you start to fall apart in terms of your?
Yeah, that's a good question. So when we're doing random sampling, we take the square root of the number of samples, right? So if you have 100, you're going to take 10 samples for each one of your decision trees. That seems to work okay. That's kind of our ballpark number, is at least 100. So you have to have 100 in both groups.
Right.
So this is now a comparison of the heat maps from an earlier version versus this new version. So here you can see that there are a lot of these kind of quantitative markers. And now over here, the expression pattern is much more binary. And so we were able to kind of accomplish this goal that we had in mind. Now, the trade-off is that we need more markers to get maximum classification accuracy, but at least the markers show this binary pattern.
So why on the first version you don't have those markers come out? Just because?
That is a great question. I'm not sure about that. Some of them are the same, but some of them are different. It could relate to, there's a dropout phenomenon in these data that just for technical reasons, these genes are not expressed in all of the cells. And so you kind of saw that in the previous diagram here. There's still variability in the expression of this guy, and some of these look like they might be technical dropouts. And so it might relate to that in some way, but I'm not 100% sure. That's a good question.
Is the purpose of these just to then be able to look at insect hybridization?
Yeah, that's one of the downstream applications. So, so now this is the 75 cell type clusters from the middle temporal gyrus. And now this is the collection of marker genes that we need to be able to tag each one of those 75 different types of cells. This is their kind of the median expression across all of the nuclei in each of the clusters. And so we need 157 marker genes for the 75 clusters. So on average between 2 and 3. And we get a pretty decent classification score when we use those set of markers. I was pretty surprised that we only needed between 2 and 3 markers because these neurons are quite similar to each other, and yet it seems like there's a couple of key genes that really distinguish between their phenotypes. So here are just a couple of examples of what these markers look like. So down here, on the very end here, this is the microglial cluster here. And you can see that there are actually 2 genes that are really, really good just by themselves in allowing us to identify microglial cells in comparison to these other cell types. That's not too surprising since most of the other cell types are neurons. So obviously microglia would be expected to be quite different. This example here I was a little puzzled by initially because I was looking at this. These are the 2 markers that are required for the first cluster. And I was like, why is this gene not enough? It seems to be very specific for this one. It's not found anywhere over here. But it turns out if you go all the way over here to the right, this gene is expressed in microglia and this one type of inhibitory neuron. And this is TGF-beta receptor. So I'm an immunologist, and I feel like TGF-beta receptor, that's got to be interesting. So TGF-beta receptor is expressed in one type of inhibitory neuron, and this is one that's on layer 1, so the very apical layer of the cortex. And so I'm wondering if there isn't TGF-beta in the cerebral spinal fluid, and maybe it's influencing the function of this neuron. So something that I'm hoping that at some point maybe we can explore experimentally. And then lastly, these are some of the excitatory clusters here. And it turns out that a lot of the marker genes, especially for excitatory neurons, are these only have locus IDs. They don't have gene names. And so these things are really, really specific for certain subsets of neurons, and we know nothing about what they are. They're completely uncharacterized. So I think there's going to be a lot of new biology that we'll be able to discover by pulling out these kinds of genes from this analysis. So, I mean, how do we know that we're really getting good marker genes from this kind of analysis? And so we did one experiment that I think is probably the most compelling argument to say that these really are good marker genes. So this was the original clustering result that I showed you earlier. This was generated using the 3,500 genes that are most highly variable across the data set. So we basically redid the clustering just with the 157 marker genes from the analysis, and we've almost identically reproduced the clustering results. So you can see here, like for example, this cluster is this cluster. Kind of hard to see the colors. This cluster is this cluster. This group is this group. You have these guys over here. You've got this group is up here. This group is here. And so with these 157 genes, we seem to have been able to completely recapitulate the cell type identity that you get from this larger transcriptomic data set. Okay, so there are 3 things that we're doing with these marker genes. We already talked about the multiplex fish. Those experiments are kind of underway now. But what I'm going to tell you about for the rest of the time is we want to generate a reference database so that when somebody does a new single nucleus RNA-seq experiment, they want to know, have these cells been identified before, or are we dealing with novel cell subsets? And then the other thing that we want to do is we want to produce a semantic representation of this to add to an ontology that we've been developing called the cell ontology, so that we can now start to have conversations about these cell types in our discourse. Renee Zhang is a staff scientist at the Venture Institute, and she's been working on the cell type matching problem. So the idea is that, you know, we do this, we pull out these marker genes, and then we want to explore the possibility of just generating a reference expression matrix that's just composed of those marker genes. And then when somebody has a new set of data, what we would do is we would pull out the expression of just those marker genes from this new data set, and then compare that pattern with our reference database. And so to do that, we are using a statistical test that's called the Friedman-Rafsky test. So the Friedman-Rafsky test it's kind of a graphical representation of multidimensional data, and it allows you to do a 2-group comparison to determine whether the distribution of these multi-dimensions is the same or not. And so the way that it works is we basically generate this completely connected graph where the distance between the nodes here are basically some distance metric. In this case, it would be the Euclidean distance. So imagine that you have a vector that's pointed in 150-dimensional space, and then you have for one nuclei, and then you have another nuclei. You can measure that distance using Euclidean distance or other distance measures. So we generate that graph here, and then from that we generate what's known as a minimum spanning tree. So we're basically just making sure that all nodes are connected by a single edge, and we want to have the smallest length of edges in this minimum spanning tree. So, and then what you do is you remove all the edges that connect nodes from the 2 different clusters that you're trying to compare. So basically, we've merged the data from the 2 clusters, generated the minimum spanning tree, and now we'll remove anytime there's an edge that connects nodes from the 2 different clusters. So when you do that, if your data from your 2 clusters is interspersed with each other, you have lots of nodes that connect nodes from the different clusters. So you end up with lots of subgraphs here. But if your clusters are separated from each other, you basically don't have you have maybe one node, one edge that connects nodes from the different clusters, and you end up with very small numbers of subgraphs. And so this is now you can calculate a statistical measure based on this kind of analysis, and to tell you whether your data is interspersed in that high dimensional space or not. So what we do then in this algorithm that we call FR match is we take the data from one cluster in one data set and data from one cluster in another data set. We generate this minimum spanning tree, and then we calculate this FR statistic and the p-value. So this is what some of these minimum spanning trees look like. So here you can see these 2 obviously are quite distinct from each other. There's only a single edge here that's connecting nodes from the different clusters. Here's another one. You can see they're really nicely segregated from each other. But in contrast, this one here, you see that they're all nodes from the different clusters are interspersed with each other. And so this is the kind of criteria that we're using to indicate whether the two clusters match each other or not. So we just finished doing a second region of the brain. We just finished doing the motor cortex, the M1 region of the brain. And so we decided to compare the cell type clusters from the motor cortex with middle temporal gyrus. So first thing is we pull out the marker genes from M1. So here's the MTG heat map that you saw before. Here's a similar heat map from M1. You can see what it's remarkable. It's like, so there were 77 cell type clusters in the motor cortex. We need 158 marker genes here instead of 157. The average is 2.4, and this F score is very similar. I don't know why 2.4 is the magic number, but we got it in a completely different brain region. So then, if you look at the expression of these marker genes in nuclei from these different clusters, you kind of get a barcode representation. So these are all the marker genes. So these are the markers for this first cluster. They're not expressed in these other clusters. But you see that they do express some of these other marker genes in this kind of stripe pattern. And then this is a set of marker genes for a second cluster, third cluster, fourth cluster. But this stripe pattern actually is kind of unique for each of these different cell type clusters. And this is what's being compared in this FR test. So basically we can compare M1 to MTG using the M1 marker genes, or we can compare MTG to M1 using the other set of marker genes. And this is the matching result that we get. And then we can also look to see where do we get two-way matches when we go in both directions. And so this is the result. Out of the 75 or so cell type clusters, we have a two-way match in 31 of those and one-way match in 18 of them. And then these are the ones where there's no matches. So we have pretty decent confidence that when we get a two-way match like this, that we are really matching to the same cell type from these two different brain regions. When we get a one-way match, we're not too sure about that yet. So this is kind of an area that we're exploring further. And then if we don't get a match, we're pretty confident that these are now distinct cell types that you see in these two different brain regions. So all of this is pretty fresh off the press. It's works in progress, but this is kind of the things that we're trying to accomplish in this project.
And these data sets are still the binary version of expression?
Yes. So now we selected these marker genes using that version 2. Okay, so then the last thing that I'll mention briefly is the semantic representation part. And this is work that was done by a bioinformatics engineer in my group, Mohammed Kesh. So we want to then use these marker genes also to come up with a textual representation of cell types. And so we work on building ontologies. And a lot of times people ask me, well, what is an ontology? Isn't that just a vocabulary, dictionary, or something like that? And the difference between a vocabulary and an ontology is that ontologies are structured vocabulary, so they have terms and they have definitions. But the thing that ontologies do that you don't find in, say, in a dictionary, in a vocabulary, is that we also capture information about the relationship between terms. So, for example, for cell types, we want to capture type-subtype relationships, and we use the ISA relation to represent that. We can capture developmental relationships between cell types using develops from relationships. We can capture information about the tissue source of the cell using kind of a part of anatomy term. And we can also capture functional information about the cell has disposition to realize certain functions. So what we want to do now is take this information that we're accumulating from these experiments and incorporate them into an ontological representation of this information. So we came up with a proposal about how we would approach this for neurons in the brain. And this should be extensible to other tissues as well. So we want to combine the specimen source description, and that would include the anatomic structure and the species, with a parent cell class, like the kind of the higher level class of cells that we're talking about. And then the necessary and sufficient marker genes that are selectively expressed in that granular cell type. And so what that might look like is, in kind of a sentence form, is that this particular type of cell is a human middle temporal gyrus cortical layer 1 GABAergic interneuron. So this is the parent cell class, and this is the anatomic region that selectively expresses these 3 genes. And so now this is a specific definition of that cell type in kind of a language form. And so then we're producing these things and incorporating them into a provisional cell ontology knowledge base, so that other people then could get access to it. And then so we can represent this using an ontology language called AL, or we can also represent it using a different representational format, RDF, which generates kind of triple assertions that you can query using this query language called Sparkle. But I'll just kind of show you an example of what this looks like. So here is, BioPortal is a public database of ontologies. And so we have the provisional cell ontology in BioPortal. And this is a representation of all the middle temporal gyrus cell types that are now here. And so here, for example, is the definition of VIP expressing humans through the cortex and G, yada, yada. We also capture information about the location of the cell body in the different cortical layers. We have information about the parent cell type, which is here. So this is the parent cell. And then, there's other kinds of information that we can also capture in this representation. So this is one way to view it. The other is you can represent this using this language called RDF. This is kind of what that representation looks like. But one of the big advantages of representing this in RDF is that then you can query this whole database using this query language called Sparkle. And this is kind of the results that you can get then is, show me all of the GABAergic and glutamatergic neurons enriched in layer 4. And these are the cell types that meet those query criteria. And this, these are just different kinds of queries that then you can launch. So all of this has been based on transcriptional profiling so far, but we are also starting to capture other information about these cells, their morphology, their electrophysiological characteristics. And so now what we're trying to do is come up with strategies of how do we represent this kind of result in a semantic representation. I can tell you it's a lot more difficult than the marker genes, but that's something that we're starting to work on now. And then the last 2 slides, I promised I would say something about immunology. So we are also, we've also started to use this 10x Genomics platform, which I'm sure most of you are familiar with. The workflow is slightly different, and I think SmartSeq2 and 10x have their advantages and disadvantages. SmartSeq2 gives us full-length transcripts, and we tend to detect more genes than we're detecting with 10x. But it's very labor-intensive, pretty expensive, and it's very difficult to analyze large numbers of cells. That experiment with 16,000 nuclei was a huge undertaking. 10x allows you to do 10,000 cells easily, but you only get sequence data from one end of the transcript or the other. And the number of genes that you detect with the 10x platform tends to be lower than what we typically get with SmartSeq2. So it just depends on your application. So anyway, we've been using 10x to look at vaccine responses. And this is kind of an example of what the clustering results look like. We get decent clusters. You could figure out what those cells correspond to by looking at marker genes. I think the one thing is that I suspect if we took these same samples and analyzed them with SmartSeq2, we might get a larger number of clusters because we're detecting more genes. But we're just actually doing a head-to-head comparison of the two technologies now, and we know what that looks like. You know, we think that this single-cell transcriptomic platform is really revolutionizing our understanding of cellular complexity. You know, in terms of human brain, you know, we're detecting probably 5 to 10 times more types of cells than we originally thought we might see based on the prior knowledge in the field. We think that the features that are useful for classifying cell types, not only can they be used as molecular biomarkers of cell types, but they frequently point to important underlying biological phenomena like that TGF-beta receptor example. So this is why we think using artificial intelligence approaches that retain explainability in terms of the features that are used for classification for modeling have some real advantages, especially in the biological field. So we have this method NS4s that's giving us this minimum set of marker genes, and we can use those genes as probes for multiplex FISH, as a condensed reference data matrix for comparing new data sets to determine if we're seeing the same or different cell types. And we can use them for generating these semantic cell type representations. And so we've got a bunch of papers that have been published. We've got a Nature paper, it's actually in press now, that describes the middle temporal gyrus experiments that I mentioned. And this has been a really terrific collaboration between our group and a group in the Allen Brain Institute in Seattle. That place is phenomenal. If you ever get a chance to go and visit them, definitely go and see. And we've been working with Ed Lean's group in the Allen Institute for Brain Science and these people in this group. And then we've had other collaborators working on different parts of the project. And this work has been funded by the Chan Zuckerberg Initiative, the Human Vaccines Project for that last little bit. And then we got some pilot funding from my institute to get us started. So that's it. Thanks for your attention. And
So when you mentioned you did the 8 samples together, some from frozen, some from fresh tissue, didn't you put the sequencing libraries together and run it on the same run?
so they were, no, they were not.
So even despite that, you didn't have much tracking.
Right, right. We certainly couldn't run all the samples in the same run because there were too many. And we like to get about 2 million reads per sample. So 2 million times 15,000. So those are the number of reads that we were trying to get. And then even doing 19 or 16,000 libraries at the same time just isn't feasible. So we definitely did them in batches.
That's just amazing that they mix so well.
Yeah, I know. I was really surprised. And we didn't do any kind of batch correction or anything on the data. But this is the advantage of working with the Allen Institute. It's like a machine up there. You know, they have all this robotic equipment and everything. So we spent 2 years developing the standard operating procedures to do the single nuclei RNAseq. Once we were really happy with, you know, how the quality of the data that we were getting, we then went into production mode. And I think that was really important to get this kind of quality data.
So clearly the transcriptome of single cell under the influence of disease is going to change. Are you still going to be able to identify it as the cell type in normal tissue?
Yeah, we're just getting started with that now. So we're collaborating with the Alzheimer's Disease Research Center here on campus. And so we just got our first set of nuclei from a couple of Alzheimer's frozen brains. And so we're going to start doing this comparison. But it's definitely going to be tricky because if you see something that's missing, is it because you just didn't sequence enough, you didn't do enough nuclei, and it's there just at a lower abundance? So we'll see. But I think we will be able to do that matching, and there will be some things that are not matched. And it's going to be tricky to figure out if that's real or not.
You're talking to a bunch of HIV researchers. Why aren't you doing HIV?
Well, I think that's why Doug wanted me to come and talk to you guys.
So with regard of the matching, like say, sorry, this is very technical, and which means under two kind of clustering methods, which means you really identify the cluster which is unique. Will be some cells or cluster in the new samples, which is actually from originally the two cluster, is like in the middle. So how that, you know, I mean, because the whole the cell development, everything change is continuous.
Yeah.
It's not
Right.
either this cell type or that cell type.
Yeah.
So how that kind of matching will be effect. So maybe you are not detecting the new cell group, just in the between transitioning.
Mm-hmm. Yeah, so this is all with adult brain. And so there's basically no differentiation occurring with this set of samples. But you're absolutely right. We're also, you know, looking at fetal brain development in other projects. And there you don't see as nice discrete cell type clusters. You see more of a continuum. And so I don't think you can apply this same kind of method when you have that kind of situation. But I suspect that what you see during development is that you have your precursor cell and you have your mature cell, that those are discrete types of cells and that there's some stability in those transcriptomes. But there will be this connection between the two as they transition from one type to the other. So I think for the beginning state and the end state, you should still be able to do this comparison, but not with the trajectory in between.
So in the immune clustering, cell clustering, you look at cell types. But for example, T cell subsets,
right?
They all cluster as a certain type of cell. Right? You don't have the maturation or differentiation subtypes.
Yeah. So I think in this case here, where we did have this subset of T cells here, one of the things that you can do in this process is you can do what's called iterative clustering. So you do an initial clustering when you get these major classes, and then you take these guys, you pull out the genes that are differentially expressed within that group, and then do a second round of clustering. And then sometimes you would get T cell subsets when you do that kind of analysis. So that's possible. But the other possibility is that it could be that using 10x, we're not getting enough of those genes that distinguish between T cell subsets to allow us to generate those clusters. So I don't know.
I'm surprised your B cell clustering is so non-specific.
I know, I am too. Yeah, you know, I was a little bit disappointed with these results, but the clusters are pretty good. And I think for the purpose of understanding the vaccine response, we think we've found some interesting biomarkers from this. So we'll see. We have to follow up on those. Yeah.
Have you looked at whether the coincidence, if you've heard of about 150-ish genes and 2.4 per class scales at all with the sample procedure? So you mentioned you were using about 100 for the number of samples. Is that number pretty robust?
I mean, we could certainly do that. You know, we haven't tried to fiddle around with our sampling strategy just because, you know, the core random forest algorithm that we use is just out of the box, and that typically is how it selects the number of samples to use. But I'm not sure. So we haven't looked at that. I hope not.
So in this comparison of SmartSeq versus 10x,
yeah,
you did the analysis for 10x using the same algorithms, the same random forest algorithm?
We will, yeah. So the processing, the initial part of the processing pipeline is different because 10x uses these UMI barcodes, whereas SmartSeq will not. So that part will be different, but as soon as we get to kind of the expression matrix level, from there forward, we'll use the same methods.
For me, because like you mentioned, next detect less genes.
Yeah.
Might be the 2.6 per se, or might not hold, is because we need more genes.
Yeah. Well, either we'll need more genes or we won't get the same level of granularity. So I think it's going to be one or the other probably. All right, well, thanks everybody. Thanks.